everyone! Today I'd like to talk to you about the importance of hormones in plants and how NMT can help you reach new levels in your research in this field of study. Similar to hormones in animals, hormones in plants, such as auxins, play a major role in growth and development. If it weren't for these hormones, plants would just be a clump of cells without form or function. Among other things, plant hormones shape the plant, affect gene expression, and even affect the sex of flowers. Despite their importance, the study of auxins has been very limited since its discovery over 130 years ago. In 1880, Charles Darwin first recognized the effect of light on movement in canary grass coleoptils. With his son, he found that these coleoptils only bend towards the light when their tips are exposed. These results suggested that the tip of the coleoptil detects the light, but that there was some messenger being transmitted to the lower part of the coleoptil which was causing it to bend. In 1885, Sulkowski first isolated IAA, the most important auxin, from plant tissues. But it wouldn't be for about 50 more years that scientists would be able to isolate it from plant tissues for a second time. It wasn't until then that IAA was officially recognized and named. Since a very low concentration of hormones is required for plant responses, it had been very difficult for scientists to study plant hormones in their natural state. But in the early 2000s, Stefano Mancuso was first able to do what other scientists could not. With his lab partners, Mancuso developed a microelectrode that was able to detect local in vivo concentrations of IAA non-invasively and in real time. This newly developed microelectrode was a breakthrough in the study of IAA physiology in plants. With help from Dr. Marshall Porterfield from Purdue University and Dr. Eric McLamore from Florida State University, YoungerUSA now provides commercialized IAA Flux sensors to scientific communities worldwide. This new flux sensor made it possible for scientists to measure the influx and efflux of IAA, which was a huge advantage to this field of study. With the use of this commercialized flux sensor, research in plant hormones has been more prominent than ever, and scientific breakthroughs occur every day. One notable breakthrough was illustrated in the Journal of Plant Growth Regulation. In 2015, Suli Yan, Ting Zhang, and others found that methyl jasmonate affects root growth by modulation of transmembrane auxin flux in the transition zone. The understanding of how IAA affects plants is only just beginning. If you are interested in plant growth and regulation, the IAA Flux sensor will undoubtedly benefit you in your research. If you are interested to learn more about the IAA Flux sensor or real-world applications of NMT, please subscribe now or visit our website. Thank you for watching.